High-resolution structural biology techniques like cryo-EM and X-ray crystallography have transformed our understanding of proteins and biomolecular complexes. But even the most advanced instruments can rescue a poor quality sample. In many cases, failed structural studies come down to one overlooked problem. The sample wasn't homogeneous, stable, or properly assembled to begin with. Welcome to Tell Us What You Know, a series from Drug Discovery News where we ask scientists to break down today's most exciting topics in research. Today, we are joined by Brenda Watt from Refeyn to explore why sample qualification is such a critical step in structural biology workflows, and how mass photometry helps researchers quickly assess sample quality under native conditions using minimal sample. We'll also discuss why these pre-screening tools are increasingly important for AI-driven protein design and drug discovery. Let's dive in. Hi, Brenda. Thank you so much for joining us today. How are you doing?
Pretty good. Thank you for having me, Alejandra.
Thank you. Well, first, can you tell us what are the biggest challenges in structural biology and why does sample quality so often determine success or failure?
So, typical methods for determining structures are X-rays, crystallography, and cryo-electron microscopy. And they're really powerful techniques that have helped determine the structures of many biological complexes and micromolecules. But both heavily rely on sample quality because to get a good quality structural data, you need to have very pure samples that are well qualified. So there's three overarching challenges for any structural study, and they all depend, again, in one way or another on sample quality. So one challenge is having a structure with enough resolution, and this is about how precisely you can map different parts of the protein, even single atoms, onto a 3D map of the structure. And ultimately, everybody wants atomic detail. But you can get much better resolution structures if your samples are homogeneous. Another challenge in structural biology is coverage. So the difficulty is, can you build a complete model? Do you know what's in your sample? Are all the components involved present, or are any of them missing? So being sure that you have what you think you should have it is a challenge. And this also relates to a third challenge in structural biology, which is, have you captured the right structure or set of structures, especially when there's flexible or dynamic regions? Are they visible or are they blurry, poorly resolved? Because many molecular machines transition between different states. So they might change conformation, they might lose or gain subunits, and it can be hard to reconstruct a full pathway when you only have snapshots of maybe only the beginning or the endpoints. So for both cryo-EM and crystallography, sample quality and heterogeneity, especially heterogeneity, is a problem. So for crystallography, you simply can't get a crystal to form if your sample is heterogeneous. And while cryo-EM can handle heterogeneity much better, if you have too much of a mix of assemblies, different stoichiometries, or different conformations, you end up either just missing out on certain species, but also kind of effectively averaging some of these species together, which results in a lower achievable resolution and sort of a blurred signal. So this is why really qualifying your samples, trying to, I know that we want to capture all these different states, but finding a way where you can have a sample that just has this state, just has this state, or simplifying the system results in much better resolved structures.
I see, thank you. And now, what do researchers typically use to qualify samples and where do those methods fall short?
Yeah, so some very widely accessible techniques to qualify samples are SDS-PAGE and size exclusion chromatography, in particular for structural studies. And both of these techniques are able to separate proteins primarily based on their size or molecular weight. So SDS-PAGE is often used to confirm which subunits are present in a sample, if there's impurities, is there fragmentation. But it's a denaturing technique. So you use heat and detergent during the treatment of the samples, and this disrupts complexes. So it can't reliably report on native stoichiometry. So for example, if you have a single component system, you might get a single band by SES page, but you don't know if this reflects a monomeric species, a tetramer, or maybe even a mix of different oligomeric states, because no matter what those states are, you would still get a single band. And likewise, if you, let's say you have a multi-component system with 3 different species, you might see all 3 of them and know that they're present, but you don't know if you have like a 1-to-1 1-to-1-to-1 ratio, a 2-to-2-to-2 ratio, you cannot distinguish that.
Yeah, yeah,
So that is one technique.
yeah,
And this is where SEC can play an important role. So this other technique, size exclusion
yeah.
chromatography, because it runs under native conditions, and depending on the resolution of the column, it can distinguish oligomeric species and separate complexes. However, it separates by an interplay of both size and shape rather than just by size alone. So different mass species with similar hydrodynamic properties can overlap with species between the species of interest and species that you're not interested in. So even though from an SEC chromatogram, you can get a nice peak that looks homogeneous, it might represent a species of similar hydrodynamic radii that are different species in terms of stoichiometry. So it's really important to recognize that that represents an average of what's eluting at the column. And then when another challenge I think that's present, so we have SDS page, is the challenge is that it's denaturing, so you can't distinguish stoichiometries. But then while SEC can separate in native conditions, if you have now again a multi-component system where there's maybe multiple peaks, it can be hard to tell which peaks are the species of interest because those reflect the combination of both mass and hydrodynamic radius. So a third analytical technique that you can add to this to get more information of your samples is to couple size exclusion chromatography to multi-angle light scattering, as this scattering information can be used to determine the mass of the molecules eluting at any given time from the column. But if your column is not separating those different species, then the mass readout that you get is really an average of the masses of those species. So again, you're still kind of blind to what is eluting from your column.
I see. Thank you. So then, what would the ideal pre-screening workflow look like before committing a sample to a cryo-EM or crystallography?
Yeah, so well, SDS page and SEC are really complementary to each other and provide really important information. They often don't paint a full picture of the sample. Again, in the case of SDS page, because it's denaturing, so it can't reveal multiple stoichiometries. And SEC because it's a bulk measurement. And in addition, both techniques use a fair bit of sample, which you might not always have a lot of, and they can be slow. And some samples are very delicate or time sensitive. So ideally, you would want a fast method that works under native conditions. So it gives you a mass readout. It reports on purity and stoichiometry. That's what's nice about mass readouts. So you can confidently choose the best sample or the best fraction for downstream structural work. And this is where single molecule techniques like mass photometry can play a really important role because single molecule techniques avoid averaging. So as we said, like maybe many different types of molecules are eluting from a column, so you're just averaging them. But by measuring the mass of single molecules in solution under native conditions, mass photometry can quickly reveal whether a sample is monodisperse, what stoichiometries are present, the relative abundances of them. So that's really powerful information. In addition, mass photometry uses very little sample, so you also don't end up having to choose between, do I have enough sample to do all the analysis I do to be sure to go to the next step? Or if I do all these analyses, now I won't have enough sample to go do structural work. So the fact that mass photometry uses so little can also be, whoof, I don't have to run out of sample before I do my next stage of the study.
I see. And then mass photometry has been around for a few years now. What's new and how does your latest instrument make the technique simple and more accessible?
Yeah. So since its commercial introduction in 2018, mass photometry has been adopted by like pretty profoundly across academic and industrial labs as a quick and information-rich check for sample quality. So this has helped researchers optimize conditions that stabilize any species that they're interested in, confirming readiness for cryo-EM or crystallography, also studying protein-protein interactions and assembly mechanisms by directly reading out stoichiometry, and the measurements just take 1 minute. However, we recently developed a new instrument we're really excited about. It's called MyMass, and it's designed to reduce friction for everyday use and broaden access. So many mass photometry users have mentioned that they often can only access the instrument in a core facility or a different lab, a different building. Sometimes they even have to go to another city to do their measurements, which is not really where they're preparing their samples. MyMass has been designed for those users in particular and is more affordable. It's a compact benchtop instrument with an integrated touchscreen and requires no warm-up time, which is also a really big plus. So users can check samples on the fly while they work on the same bench that they're doing their sample preparations, and this allows them to make decisions in real time.
Thank you. Finally, as artificial intelligence transforms protein structure prediction and protein design, why is experimental validation still so important?
That's a really good question because in silico models are becoming incredibly powerful for proposing structures, exploring alternative states, and even guiding the design of new proteins and therapeutics. They can help us generate hypotheses about intermediates that are hard to capture experimentally, and also prioritize which constructs are worth pursuing and taking further. But they still need to be checked against experimental reality. So model performance depends on the quality, quantity, and diversity of the underlying data. And there's regimes where the data are effectively shallow. So for example, when you're designing sequences that don't resemble a natural protein, so you don't have natural protein structures to kind of inform onto these novel proteins that you're trying to design. Another case is when complexes are really flexible or populate multiple states in solution, because training data is often biased towards the most stable conformation or structures that have been captured under very specific experimental conditions, like crystal packing. But these might not reflect what the protein does in solution or in cells. So I think we need to build more draining data that has more in solution information uh, because a lot of the information that we have has been built on, for example, crystal structures that showcase maybe the most stable conformation a crystal can be packed in, but not the reality in a cell, which is in solution. So I think this is where a fast, low sample consumption experimental checks become really important. So AI can give you like a whole list of candidates, and then it can also help you narrow down which are the best ones. But then you need a fast, low sample consumption experimental check to confirm that you've actually had the intended species. So it has the right assembly, it has the right stoichiometry, and it's not aggregating. So once you've made these checks, you feel much more confident to invest in downstream structural work. But you also don't want to make a whole bunch of protein for maybe a still pretty big list of candidates and do a lot of analytical development and method development to analyze a whole bunch of structures or species or samples that you don't really want to invest time in. So mass photometry is a really good fit at this stage because it's quick, it works under native conditions, it uses very little sample, and there's like no method development. So it gives you a direct readout of purity and oligomeric data and stoichiometry information. And we hope that over time, this kind of information can also help close the loop by generating or providing information to generate higher quality experimental data sets, which have this oligomeric state or stoichiometric state information that is so hard to get from other techniques. And hopefully that also improves future in silico models. That's the aim.
Yeah, great. This was incredibly insightful. Thank you so much for sharing your expertise with us, Brenda.
Thank you, Alejandra.
If you enjoyed this episode, be sure to like, subscribe, and check out our other videos for more expert insights on science. Thanks for watching. We'll see you next time.